Let us open up your box with supplies and see what's in there. The most important thing in the box is, of course, the microscope. Take it out and carefully place it on a table or other solid surface. Do not throw away the microscope box and styrofoam container. You will need them to return the microscope at the end of the semester. There will be a separate video on the use of the microscope. There is a sleeve with 15 plates of trypticase soy agar, TSA, labeled with a pink line down the side of the plates. There are also 4 plates of Mitis salivarius agar with telluride, MSAT, and these are bright blue in color. There are 4 plates of potato dextrose agar, PDA, which are marked with a dark purple line. You also have 1 McConkey agar, MAC plate, burgundy in color. And lastly, there is 1 blood agar plate, bright red in color. Of all the plates you received, the blood agar plate is the only one we purchased commercially. If you turn the plate over, you will see black letters stamped on the bottom of the plate. All others were prepared in the Santa Fe College Microbiology Laboratory. You have a tube rack with 4 conical tubes with media. Sadly, the 3D printed rack is a bit fragile. The legs come off easily, so be careful when you handle it. 3 of the tubes have liquid media, broths. They are PR glucose, PR mannitol, and trypticase soy broth, or TSB. There is 1 agar deep in the rack, thioglycolate agar. This is a jelly-like agar which should be handled carefully so as not to upset the oxygen gradient in the tube. There are 2 small tubes, one with crystal violet and another with saffron in. The rest of your supplies are in the plastic bag. Let us check out all of these goodies. In the bag, there is a conical tube with sterile water and an agar deep with a medium called SIM. You have a slide box with a lid filled with 10 glass slides. If you are careful, you can reuse each slide multiple times. I will post a separate video on how to clean them after use. You have a small container with cover slips. We only use cover slips for the wet mount. You should have about 4 of them. Note that they are separated by sheets of paper to help keep them apart. You will only need the transparent cover slips. The paper may be discarded. For performing bacterial staining experiments, you have a slide holder and a waste cup. There is a groove in the holder where the glass slide fits in. Once the slide is in, it will fit right on top of the provided 5-ounce waste cup. There is a supply of inoculation loops, blue in color. If you unwrap this carefully, you can take the loops out one by one without exposing any others. The same is true for the inoculation needles, purple in color. There is a small magnifying glass, a keychain UV light. Remember not to shine the UV light in your eyes, it can damage the retina. A sterile cell spreader, cotton swabs, transfer pipettes, forceps, sterile toothpicks, a thermometer, lens paper, bibulous paper, a Sharpie, an ELISA strip, and a cotton ball. You have an empty dropper bottle to put isopropanol in. You will need that for doing the gram stain. A brown bottle with grams iodine, a little plastic bag with glow germ powder, and a small envelope with some yeast granules. There's also a small tube with blank paper discs in them. And wrapped in aluminum foil, there are sterile Eppendorf tubes. Be sure not to open that until we need them. We will use those if and when the pipetters come in later this semester. And there you go, everything you need for a semester of Microlab at home.